Next class is fatty acid oxidation. This fatty acid oxidation is nothing but degradation of fatty acids. In the previous lecture, we had the different pathways leading to the synthesis of fatty acid. Now this is fatty acid oxidation. The fatty acid oxidation is a very important step involved in the biochemical pathways and the biochemical metabolism. And this is the introduction of beta oxidation. The oxidation of long chain fatty acids to acetyl coenzyme A, from the fatty acids to acetyl coenzyme A. Previously we had an outlook of acetyl coenzyme A converted into malonyl coenzyme A, and malonyl coenzyme A subsequent elongation leads to the complete fatty acid synthesis into C16 carbon, that is palmitic acid, and palmitic acid further elongated into different fatty acids including arachidonic acid, that is maximum chain containing carbon containing fatty acid. So now we are going to chop each and every 2 carbon from the fatty acid, and this 2 carbon will be formed by combining with the coenzyme A. The acetate moiety combining with the coenzyme A, this is called as acetyl coenzyme A, will be produced. So it's a central energy yielding pathway in the mammalian heart and liver, and it provides 80% of the energy need. So this oxidation is a very important component, maybe all the animal system, mammalian system, and depending on this beta oxidation for the energy production. The fatty acids are converted into acetyl coenzyme A by a repetitive 4-step process. So only 4 steps are available. It's called as beta oxidation, and the beta oxidation means the combining of all the 4 steps is called as beta oxidation. The advantage of fatty acid as a storage fuel, that is highly reduced structure with high energy, because the fatty acids are the high energy substances, and maybe it is very easy to store in the adipose tissue. So that is why we are storing the fatty acid as a storage fuel. At the same time, if there is a need of energy, and if the particular subject, the mammalian subject, maybe in the starvation process, automatically the storage fuel is utilized as an energy substance, and that will be subsequently yielding acetyl coenzyme A, and this acetyl coenzyme A enter into the TCA cycle and produces NADH, and this NADH subsequently will enter into the electron transport chain for the formation of ATP. And this will not raise the osmolarity of the cell, and there is no disturbance in the physical nature of the cell also. So undesired chemical reaction with other cellular constituents, it is not possible. There is no undesired chemical reaction is possible. That means ultimately the very efficient product of storage is the fatty acids. So that is why we store fatty acids in our adipose tissues. The fatty acid oxidation happens in mitochondria. The location or localization of all the fatty acid oxidation, the beta oxidation, happens in mitochondria only, in animal cells. And the enzymes of fatty acid oxidation in animal cells are located in mitochondrial matrix. If you're asking the question where it is located within mitochondria, means it is located within the mitochondrial matrix, that is a liquid or the watery substance present within the mitochondria. In mitochondria we have outer membrane, inner membrane, and the cristae, the finger like the protrusions within the mitochondria. It's called as cristae. And this one is mitochondrial matrix, the liquid portion, and the aqueous portion of mitochondrial matrix that is involved in the fatty acid oxidation. So the fatty acids are activated before entering into the beta oxidation process. You can say that, what is the meaning of activation? Activation means only the fatty acids will not enter into the fatty acid oxidation. So for the entering into the oxidation, we need the attachment of coenzyme A. Coenzyme A is a coenzyme default, that is going to be attached with a fatty acyl moiety of all the different types of fatty acids, and this fatty acetyl-CoA moiety is the active one, and that will enter into the beta oxidation process For example, okay, this one is ATP attached with 3 phosphates. This is ATP, adenosine triphosphate, and RCOO is the fatty acid. Okay, this is the fatty acid, and the fatty acetyl-CoA synthase enzyme. The fatty acetyl-CoA means the fatty acyl moiety attached with the coenzyme. This coenzyme attachment is done by an enzyme, very important enzyme, that is called as fatty acetyl-CoA synthase. So the synthase will attach initially a phosphate and from adenosine moiety. So you can see here, okay, this adenosine with one phosphate is going to be attached here. At the same time, okay, the next step, this AMP is released. In the place of AMP, the coenzyme A is going to be attached, because this is a high energy bond. That is why this energy given by ATP. So ATP is driving the force, and ATP is donating the energy, and this energy is required to attach a CoA molecule. So that is why, so this is energy driven pathway, and the energy driven reaction, and this ATP is involved here, and ATP is converted to AMP, and further this coenzyme A molecule going to be attached with the fatty acyl moiety. The finally fatty acetyl-CoA is produced. Okay, it is given in this picture. Next one is the transport of fatty acyl moiety into the mitochondria, because this this reaction happens in cytosol, and this has to be carried out or carried to the mitochondrial matrix, because the mitochondrial matrix, you can see here, this is outside the fatty acetyl-CoA is present, but inside, okay, this is the matrix of mitochondria. The matrix of mitochondria having all the enzymes of beta oxidation, and this is carried or transported to this place. So we are zooming, okay, this part. So this is outer membrane and the inner membrane. You can see here, this is outer membrane and this is inner membrane, and this pink color indicates the matrix. So this one is the matrix, and this light blue, pale blue color indicates the cytosol. This is the cytosol. So by zooming, okay, this part, this shows cytosol. Here the fatty acetyl-CoA preformed, okay, in this reaction, okay, is present here. Okay, this one is present here, and there is a transport system is available, okay, in the outer mitochondrial membrane, okay, that is called as carnitine acyltransferase 1. So this carnitine acyltransferase 1 is a very important acyltransferase enzyme, okay, that will actively transport the acyl moiety into the mitochondrial matrix, okay, through the intermembrane space. So this is called as intermembrane space. This is outer membrane and this is inner membrane, and between this there is some spaces there. Okay, this intermembrane space is responsible, okay, this for many of the reactions, including the electron transport chain, because you know very well, okay, in the electron transport chain, all the protons are pumped into the intermembrane space, and this is driving the force, the mechanical force for the rotation of FO-F1 ATPase. Okay, this one, the carnitine acyltransferase, just the carnitine is a substance, okay, it's organic carbon substance. So this is very, very important for the normal functioning of the lipid metabolism, and the deficiency of this carnitine causes abnormality in fatty acid transport system, and the fatty acid accumulation may happen, okay, in cytosol, and the beta oxidation is not properly functioning, okay, in the particular persons. The carnitine is maybe it's a substance derived from our dietary sources, and it is not produced by our own. And the carnitine, without carnitine, okay, there is no beta oxidation process. So this carnitine combined with the fatty acyl-CoA molecule, and this coenzyme A is liberated, and this one done by, okay, catalyzed by carnitine acyltransferase. And this carnitine attached with the fatty acyl moiety is now transported through another transporter. Okay, this is called as carnitine acyltransferase 2. Okay, this another enzyme, okay, that is called as a transport protein, having a channel, that is a small channel, is port is there. Through the port, okay, this is going to be transferred or transported, okay, inside of this matrix. Now you can see here. So this is carnitine acyltransferase 2 with a transport protein. Okay, both are attached. So this transport protein is activated only because of, okay, this carnitine acyltransferase 2, and through this the carnitine fatty acyl moiety is transported, and the main function of the transport only to transport, okay, not for converting the acyl carnitine to carnitine and acyl moiety. It is not like that, okay. Maybe this is done by the enzyme carnitine acyl transferase 2, okay. This is carnitine acyl transferase 1, very in short form abbreviation it is called as CAT1, and this is CAT2. So this CAT2 will be converting, okay, this carnitine acyl moiety to carnitine separately, and this carnitine is easily transported again back, okay, back to the membrane, okay, that is outer mitochondrial membrane, and ready for this reaction, that is carnitine acyl transferase 1 reaction. So there is no loss of carnitine. The carnitine, once if it is needed, okay, maybe if it is from the dietary sources, until some point, okay, maybe it is reused subsequently, no issues and all. But at the same time, okay, the carnitine deficiency will play a major role in the abnormality in the fatty acid metabolism. And this carnitine is liberated, and after the liberation, this fatty acid moiety again attached with the coenzyme A. So now this carnitine acyl transferase 2 ligate, okay, that is attached coenzyme A moiety to the acyl moiety. Now this fatty acetyl-CoA moiety is inside the mitochondrial matrix and ready for beta oxidation. So this is called as fatty acid transport within the mitochondria. This is a very, very important reaction. This may be asked in the section B also. And this is the overall the stages, different stages of beta oxidation. You can see here, this is the first stage is the removal of each and every 2 carbon into the different acetyl coenzyme A molecules by beta oxidation, and this acetyl coenzyme A enter into the TCA cycle, that is citric acid cycle or the Krebs cycle, forms 16 numbers of carbon dioxide, and 64 electrons are formed. All the 64 electrons are pumped into the electron transport chain and finally forming ATP and from ADP plus PI. Okay, this is called as electron transport chain. So this beta oxidation is a very, very important one for the formation of ATP. Ultimately, the ATP is the energy rich substance, and that is the currency of the cell. And this stage 1 is beta oxidation. We are going to see what are the steps involved and how each and every 2 carbons are removed. You can see here, this is approximately 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This is palmitic acid. And this palmitic acid is chopped into 8 numbers of acetyl coenzyme A molecules, and 8 numbers of acetyl coenzyme A molecules further enter into the citric acid cycle for the complete metabolism and for the ATP formation. There are 3 stages. Just now I said, okay, the fatty acids undergo oxidative removal of successive 2 carbon units by beta oxidation, and acetyl coenzyme A are oxidized to carbon dioxide by citric acid cycle, and third one is ATP formation. And all the overview is over. Now we are coming to the very, very important section of the fatty acid metabolism, that is beta oxidation. So you can see here, this is the beta oxidation, and C14, for example, if the total carbon is C14 and each and every time, okay, 2 carbons of C14 is removed. If 2 carbons removed as acetyl coenzyme A, remaining is C12, and 2 carbons removed is C10, and 2 carbons removed means C8, and 2 carbons removed means C6, and again further 2 carbons removed means again another C4, and ultimately 2 carbons removed means the final end product is acetyl coenzyme A. So one condition apply here, okay, if it is even number carbons and without any saturation, okay, so that is the desaturation. If there is no double bond, so this is the default pathway, okay, for the production of ATP, sorry, acetyl coenzyme A. And another condition is there, if the carbon total number is instead of 14, if it is C15 or C17, if it is C19, okay, what will be the case? So that we have to study in a different pathway. And one more thing is there, that is if it is saturated, okay, like this one, if it is there, okay, no issues, because the double bond is not there, okay, there is no much complex system, okay, for the 4 reactions for the formation of acetyl coenzyme A. At the same time, if the double bond is present here, so automatically that may be the complex one that need special enzymes to that is saturate, okay, that one, and after further saturation only this will be processed, the beta oxidation. So as a PG level students, okay, you should have the difference of the different complexity of the fatty acids. The fatty acids are classified into saturated fatty acids and unsaturated fatty acids, odd number chain containing fatty acids and even number chain containing fatty acids. Now we can complete the saturated fatty acids and even number chain containing fatty acids as a simple one. Then we can go to the next level. The first one is here. This is fatty acetyl-CoA, the polymethyl-CoA, having total 16 number of carbon atoms. Very first reaction is acetyl-CoA dehydrogenase. So this acetyl-CoA dehydrogenase involves the usage of a coenzyme that is called as FAD. This FAD is converted to FADH2. That means this is receiving 2 hydrogens. So from which part? Okay, that is from, so this the number 1, this is CO, that is number 1, this is alpha carbon and this is beta carbon. So this is happening in the alpha carbon. Okay, this is alpha carbon, means you can see here the COA is will be the intact one, and if you are removing one hydrogen from here and another hydrogen from the beta, alpha 1 and the beta 1, okay, it is called as, okay, this alpha 1, if one hydrogen is removed, okay, that is, okay, this CH, and another one is another CH, okay, from beta carbon. This is alpha carbon and this is beta carbon. So this is called as trans delta 2 enoyl CoA. Delta 2 means the second carbon atom, okay, this the delta 2 moiety. So this is the first carbon and this is the second carbon. From the second carbon to third carbon only, okay, this double bond is present. Delta 2 means second carbon to, previously, okay, we had, okay, in the that is the formation of saturation, and from the palmitic acid we had this one as an idea, okay, that is a delta 2. Delta 2 represents the double bond formed between 2 and 3. Just you can see here, number 1, number 2, this is number 3. So from 2 and 3 the double bond is there. That is why this is called as trans delta 2 enoyl CoA. So double bond is represented as enoyl CoA. Okay. And next one is enoyl CoA hydratase. Hydratase means addition of water based hydrolysis, and this water hydrolyzed to OH and H. Okay, that means the OH is placed in this place, okay, that is OH, and hydrogen is attached to here, that is the second carbon. That means CH is converted into CH2. So this will receive one hydrogen and this will receive one hydroxyl moiety here. So that is why this CH is converted into CHOH and this CH is converted into CH2. So that is a simple one. Hydratase means addition of, okay, the water moiety. Water moiety is water is dissociated into OH and H plus. So this next reaction is, so this compound is called as beta hydroxy acyl-CoA. Beta hydroxy acyl-CoA means this is alpha carbon and this is beta carbon. I already told this is, you can see here, this is beta carbon. Beta hydroxy acyl-CoA. And beta hydroxy acyl-CoA dehydrogenase. This dehydrogenation means removal of hydrogen. Okay, this NAD is a cofactor, NAD plus, converted to NADH plus H plus, and NADH, this is one hydrogen and this is another hydrogen received from here. So how it is received, we are going to see that. So this beta carbon, you can see here, this beta carbon, one hydrogen is this one and another hydrogen is from OH. And after the removal of 2 hydrogens, the remaining fragment is CO. Okay, this is the product beta keto acyl-CoA. Okay, so that is why it is called as a beta oxidation. This oxidation happens, removal of hydrogen is equal to oxidation, and addition of hydrogen is reduction. Okay, you should remember, okay, this basic principle of oxidation and reduction. So why this is called as a beta oxidation means the oxidation happens in the beta carbon. So this carbon is responsible, that is why the oxidation, that is the removal of 2 hydrogen is responsible, and the 2 hydrogens are removed as NADH plus H plus. This energy rich source and directly entering into the electron transport chain for the energy production. And beta hydroxy acyl-CoA dehydrogenase, this is the driving the force of this reaction. And further subsequently, the next acyl-CoA acyl transferase. The coenzyme A is going to come inside. Okay, that is this is acetyl coenzyme A acetyl transferase enzyme, that is simply called as thiolase. Thiolase means, okay, with the help of sulfur that is going to splice a bond. So that is why it is called as thiolase. Thiol means sulfur. Thiol based means, okay, the thiolase means, okay, based on sulfur, okay, this bond splicing of a bond or the bond breakage, okay, may happen. So this coenzyme A is so going to be attached with, okay, this CO. You can see here this bond is going to be broken down. Okay, the splicing of this bond, okay, gives CH3 CO SCOA. This is called as acetyl coenzyme A, and from the C16 two carbons are removed. These two carbons are removed in the form of acetyl coenzyme A, means the remaining portions of C14, okay, this is called as myristoyl-CoA. Okay, this is the total reaction, and all the four reactions are repetitive. Okay, just you can see here this dehydrogenase, hydratase, dehydrogenase, thiolase. So dehydrogenase, dehydratase, and dehydrogenase and thiolase. So this is a cyclic reaction, and ultimately this will form the acetyl coenzyme A if it is a even number chain fatty acid, if it is a saturated and even number chain fatty acid. So that's all about beta oxidation, and this is very important for the examination point of view. And next one is coming to the oxidation of unsaturated fatty acid. Just now I said, okay, saturated means, okay, this one is saturated. You can see here there is no double bond here. Okay, next one is saturation, either mono unsaturation or okay, the poly unsaturation. Maybe the mono means only one, maybe double bond will be there for mono unsaturation, and the poly means, okay, this more number. Okay, that may see the next step. Okay, the poly unsaturated fatty acid. The first one is the oxidation of unsaturated fatty acid. You can see here this is delta 9. Delta 9 means between 9 and 10. Okay, this double bond is present. The beta oxidation will happen. Maybe this is very easy for till this step. The 2 carbon will be removed, another 2 carbon, another 2 carbon. Okay, till this step. Okay, this will the beta oxidation. Okay, beta oxidation means that these steps will go on. Okay, till the double bond, and the 3 acetyl coenzyme A molecules are removed. So the total number of carbon atom is C18 carbon. Okay, and you can see here this 3 molecules of acetyl coenzyme are removed, and this step is stopped because due to the double bond. So here there is an enzyme delta 3 delta 2 enoyl-CoA isomerase. So delta 3 delta 2 enoyl-CoA isomerase. So that means, okay, this double bond, you can see here this is the carbon 1, 2 and 3, delta 3, okay, and 2 delta 2, okay, 1 and 2, okay, this one is 2. So delta 3 is going to be transferred to delta 2. That means 1, 2 and 3, this is delta 3. That means delta 3 means between 3 and 4, okay, this one will be there, that is a double bond is there. So that is going to be converted to, so this is carbon 1 and carbon 2, this is delta 2. Now this delta 3 is converted to delta 2, okay, by the enzyme isomerase. So this double bond nature transferred from delta 3 to delta 2. That is why it is called as delta 3 delta 2 enoyl-CoA isomerase. So the product is cis delta 3 disenoyl-CoA, and this one is a reversible step. And once if it is formed, okay, in the delta 2, automatically the beta oxidation is activated. The beta oxidation, this will go on and produce 6 numbers of acetyl coenzyme A. If it is delta 3, this will be stopped, and if it is delta 2, there is no issues. So by this way, and the mono unsaturated fatty acid will enter into the beta oxidation. Next one is polyunsaturated, oxidation of unsaturated fatty acid, and the poly means a different more number of more than one double bond. You can see here this is an example, and beta oxidation, and maybe we can zoom it, I think. Yeah, just you can have a look. So this is delta 9 and this is delta 12. So till this delta 9 1, 2 and 3, okay, 3 numbers of acetyl coenzyme A may be formed by 3 cycles of beta oxidation. So this is so called, okay, linoleal-CoA, and this one again, the previously we had an interaction, okay, that is delta 3 to delta 2 enoyl-CoA isomerase, the previous one. the same thing, delta 3 is converted to delta 2. Now this is delta 2. So now the delta 2 will carry on, the beta oxidation one cycle will go on and forms one acetyl coenzyme A, because again another double bond will be there. So further this double bond, okay, again this double bond will be further converted into 2,4-dienoyl-CoA reductase. So this will be reduced, okay, to delta 3 to delta 4, okay, and this is called as trans delta 2 cis delta 6, okay, will be this product. And further by the action of beta oxidation, the product is trans delta 2 cis delta 4 will be formed, and from here trans delta 3 will be formed. You can see here this is trans delta 3, so this is trans delta 2 and delta 4, okay, converted to trans delta 3. And next one is enoyl-CoA isomerase. This isomerase, okay, will be converting into enoyl-CoA isomerase, means okay, the delta 3 will be converted to delta 2 automatically. If it is delta 2 means, so this will enter into the beta oxidation, and 4 cycles of beta oxidation will form 5 numbers of acetyl coenzyme A, because total number of carbon is 10, and 10 divided by 2, each and every carbon is of acetyl coenzyme A is 2, and if it is there, 5 acetyl coenzyme A will be formed. Okay, I think I explained, okay, in detail, okay, everything. So again I am repeating, okay, this one, this is a little complex initially, okay, this is delta 9 and delta 12, and 3 molecules of acetyl coenzyme A are removed, okay, by the beta oxidation till this step, and remaining thing is, okay, this one, and because due to the double bond this will not carry on, okay, that is why next reaction is this product is called as cis delta 3 and delta 6. So this cis delta 3 and delta 6 will be converted to trans delta 2 then delta 6. So the 3 will be converted to 2, so that is why the further beta oxidation will happen only one cycle, because that will remove only one acetyl coenzyme A. If one acetyl coenzyme A removed means from the 12 one acetyl coenzyme A removed means remaining is 10. So this is called as trans delta 2 cis delta 4. Now this cis delta 4 should be transferred to delta 3 by isomerase reductase enzyme, and further by isomerase, okay, this will be forming trans delta 3. And again the trans delta 3 is converted to trans delta 2, and trans delta 2 is quite easy to enter into the beta oxidation. The further beta oxidation of 10 carbon and divided by 2, this 5 numbers of acetyl coenzyme A will be formed. So with this we can complete oxidation of unsaturated fatty acids. Okay, this is a polyunsaturated. Next, coming to the odd number fatty acids of beta oxidation. So we already had the previously, all these are 18, 12, 12, 12, or including this one, you can see here the C16. So this is C16, and here C18, and here C18 also. Now coming to the peculiar type, okay, this odd number chain fatty acids. The odd number chain, okay, the last carbon, 3 carbons, okay, will be formed as propionyl-CoA. For example, if you have C17 means instead of 8 numbers of fatty acetyl coenzyme removed, only 7 will be removed, and remaining is, instead of the end product propionyl, instead of acetyl coenzyme A propionyl-CoA will be formed, a 3 carbon one. And the fate of this 3 carbon product, 3 carbon propionyl-CoA, by the action of propionyl-CoA carboxylase means addition of carbon dioxide. Okay, if it is a carbon dioxide, you should remember, okay, there is a cofactor involved as biotin, and this is energy driven mechanism, because carbon dioxide is not easily attached with biotin. It needs some energy that is driven by ATP to ADP plus PI, and bicarbonate ion is donating carbon dioxide to biotin, and biotin donates or transfers carbon dioxide to this moiety, and you can see here the second carbon is going to attach with the carbon moiety. So this is called as a methyl malonyl-CoA. So this is methyl malonyl-CoA, the substance, and this one methyl malonyl-CoA epimerase. Epimerase means you can see here this coenzyme A is maybe changed into epimer. Epimer means, okay, this is the same, okay, that is CH3, okay, will be here, and the remaining thing is this COSCO. This one, that is COSCOA, okay, this one is there. So that is coming to this region, and this is going to the bottom region, okay, that is why it is called as epimerase. Okay, so by epimerase reaction, the methyl malonyl-CoA will be formed, L-methyl malonyl-CoA will be formed by the action of the coenzyme B12 and by the enzyme mutase. Mutase means the shifting of the substance, the shifting of the elements. Here you can see here the hydrogen is present here, and the remaining portion COA and SCOA will be present in the second carbon. Now this mutase means, okay, this is going to the first carbon and this is coming to the second carbon. That is why this is called as succinyl-CoA. You know very well succinyl-CoA is an end product of, one of the intermediary metabolite of TCA cycle. You know very well. A TCA cycle is comprised of acetyl-coenzyme A combined with oxaloacetate, forming citrate, cis-acetonate, isocitrate, alpha-ketoglutarate, succinyl-CoA. So this is succinyl-CoA. So succinyl-CoA is one of the intermediary metabolite of TCA cycle, and so this is the reaction, the detailed reaction of succinyl-CoA formation, and succinyl-CoA will enter into the TCA cycle. That is why, so this one is completely metabolized. So don't forget the odd number chain, all the odd number chain fatty acids, the end product of 3 carbon is the fragment will be formed. The 3 carbon fragment is propanol-CoA, and by the action of biotin, the carbon dioxide going to be attached with the second carbon. This is called as methyl malonyl-CoA, and methyl malonyl-CoA epimerase is going to be acting on this one and forming L-methyl malonyl-CoA. This is D-methyl malonyl-CoA, and this is L-methyl. Okay, L-methyl malonyl-CoA, and methyl malonyl-CoA mutase in the presence of coenzyme B12, so that is forming succinyl-CoA. Succinyl-CoA will enter into the TCA cycle. So with this we can complete all the beta oxidation and odd number, even number, saturated, unsaturated, everything. Now coming to the beta oxidation regulation of beta oxidation, and the 3-step process, okay, that is the carnitine shuttle is rate limiting for fatty acid oxidation, and the first point. Second point is malonyl-CoA. Malonyl-CoA, the first intermediate of cytosolic biosynthesis of fatty acid from acetyl-coenzyme A, inhibits carnitine acyltransferase 1. So this carnitine acyltransferase 1 is inhibited by malonyl coenzyme A, and NAD plus ratio is high, means, okay, the beta hydroxy-CoA dehydrogenase is inhibited. So this NADH levels are very high, means, okay, there is no need to produce NADH. That is why this enzyme will be stopped. Subsequently this beta oxidation is stopped, and high concentrations of acetyl-coenzyme A inhibit thiolase. If already acetyl-coenzyme A is there, means why we have to do acetyl-coenzyme A formation by thiolase enzyme. Okay, that is why it is called as feedback inhibition or product inhibition. And if the AMP is high, AMPK is activated, and phosphorylated acetyl-CoA, it phosphorylates acetyl-coenzyme A carboxylase, and which catalyzes the formation of malonyl coenzyme A, thereby decreasing the malonyl coenzyme A concentration and allowing the further process of beta oxidation. And this is the 5th step of regulation. And the 6th one is the transcription factors turn on the synthesis of proteins for the lipid catabolism. So that is PPAR alpha, okay, that is acting in muscle, adipose tissue and liver to turn on a set of genes responsible or very essential for the fatty acid oxidation. So the PPAR alpha is very important, that present in muscle and adipose tissue. That is active, maybe the parts of the body involved in the fatty acid oxidation. And you can see here the another one is a glucagon. The glucagon is a hormone, and that will activate cyclic AMP formation, and it will form a CREB formation, and this CREB2 turn on all set of genes responsible or essential for the fatty acid degradation. So glucagon is a very key important factor. Okay, there is a hormone involved in the fatty acid degradation. Okay, so with this we can conclude the fatty acid oxidation. And next part is the coordinated regulation of fatty acid synthesis and breakdown. So we already completed fatty acid synthesis. This portion is fatty acid synthesis, and this portion is fatty acid oxidation. That is why it is shown in mitochondria, and this is in cytosol. You can see here, okay, this one, and the interacting substance is malonyl coenzyme A. So this malonyl coenzyme A, this will inhibit carnitine acyltransferase 1. If the malonyl coenzyme A concentration is very high, automatically that means that has to produce fatty acids. If the levels are very low only, okay, this will be activated. That means the fatty acetyl-CoA is transported into the fatty acyl carnitine and also fatty acetyl-CoA inside of mitochondria. This is happening only because of CAT1. CAT1 means this CAT1 is controlled entirely by malonyl coenzyme A. And one more thing is the high blood glucose levels. Maybe if the high blood glucose levels are there, means immediately after the digestion process, okay, this will induce insulin secretion, and this insulin secretion, I already told, okay, there is a phosphatase of ACC, that is carboxylase enzyme, okay, will be activated, okay, by this one, and glucagon, the pKa will be activated, okay, here, okay, this is low glucose level, okay, and these are the key points involved in the regulation process. And the first one triggers the release of insulin, and second one, insulin dependent protein phosphatase dephosphorylate the ACC and activating it. The ACC made nothing but, okay, this is acetyl-CoA carboxylase. We already studied, okay, acetyl-CoA carboxylase by biotin enzyme. That is simply the phosphorylation is activating, the dephosphorylation is insulin dependent protein phosphatase dephosphorylase ATP ACC, that is why activating it. The phosphate is removed means, okay, that is why that is activation. You can see here, this is the phosphate is removed, okay, by insulin, and again this is acetyl coenzyme A to malonyl coenzyme A formed. This pKa will form ACC to ACC plus phosphate, that is phosphorylation of this one. Okay, these are the two opposite functions of insulin and glucagon. And the third is ACC catalyzes the formation of malonyl coenzyme A. This is ACC. Well, formation of malonyl coenzyme A will be formed. Malonyl coenzyme A, and the fourth one is malonyl coenzyme A inhibit the carnitine acyltransferase. This malonyl coenzyme A inhibits the carnitine acyltransferase. Okay, and the fifth one is the glucagon activates cyclic AMP dependent protein kinase pKa. The fifth one. So this low glucose level that increases the glucagon secretion, and glucagon secretion, okay, this pKa will be activated, the protein kinase. The protein kinase means addition of phosphate. The ACC attached with phosphate, and automatically this is now inactive form. You can see here ACC with a star like something, okay, that is activated form, and this is inactive form. Okay. And the sixth is phosphorylates and inactivates ACC. That is, seventh point is the fatty acids enter into the mitochondrial matrix and became the major fuel, okay, for the production of acetyl coenzyme A. So this is the complete coordination. Coordination means how a synthesis and degradation is highly coordinated with the help of hormones and some of the end products like malonyl coenzyme A. So this is a very, very unique nature of fatty acid synthesis and breakdown, and it is perfectly and highly precisely coordinated. So if any disturbance in this may create a lot of confusion, chaos in the metabolism. That is why all the diseases may happen related to fatty acids. Next, coming to the peroxisomes and peroxisomal maybe the beta oxidation in plants. You already told, okay, the beta oxidation is not happening in mitochondria in animal cells. Okay, this is in plants. So the same type of metabolism, same type of reaction, but very few differences will be there. Okay, the peroxisomes having a special type of peroxidase. Okay, this is H2O2. So O2 will be converted into H2O2. So this is the only difference. So this FADH2 is converted to FAD by a respiratory chain. That is, it is the second complex of the electron transport chain. So that is why the respiratory chain is involved for the formation of ATP. So here you can see here instead of that ATP involvement, here there is no ATP synthesis. Here there is no electron transport chain in peroxisomes. So that is why the peroxidase enzyme is involved, O2 to H2O2. Okay. This is one difference, and another difference is this one: NADH exported for reoxidation. That means, so here NAD to NADH will be formed, and NADH is exported. Exported means this is not metabolized in peroxisome. This will be transported to mitochondria, and mitochondria metabolizes NADH to NAD, and this NAD again re-enters and used for this reaction. That is why it is NADH exported for reoxidation, and ultimately, so both will form acetyl-coenzyme A, and acetyl-coenzyme A is not utilized for citric acid cycle because citric acid cycle is not happening here. There is no citric acid cycle. But in the case of animal cells, the mitochondria, the acetyl-coenzyme A enter into the citric acid cycle directly. So this has to be exported to mitochondria. That's all. So these are the differences between the mitochondria and the peroxisomes or glyoxysomes of plants. The plant peroxisome and the glyoxysome use acetyl-coenzyme A from beta oxidation as a biosynthetic precursor. You can see here, okay, this is seed triacylglycerol. You can the seed is a storage form of energy. Okay, there is a lipid rich seed. That is why we are crushing the seeds and getting all the fatty acids. And this seed triacylglycerols, by the action of lipases that form fatty acids, and all the fatty acids undergo beta oxidation form acetyl-coenzyme A, and all the acetyl-coenzyme A by the glyoxylate cycle that will produce oxaloacetate, and oxaloacetate by the action of the gluconeogenesis forms glucose. So this is called as completely gluconeogenesis. The stored lipid is converted to glucose, and glucose is used for the metabolic intermediates of the different metabolic pathways, and nucleotide synthesis by HMP shunt pathway, and glucose to TCA cycle, and TCA cycle to electron transport chain for energy production, and glucose to amino acid production, and glucose to sucrose and polysaccharide production for the biosynthesis of plant cell wall components. So this is a way how the seed stored form of seed triacylglycerols are converted into glucose, and how the glucose is utilized for different metabolic pathways. So depicted here. So next, coming to the beta oxidation is over, all the types, and the regulation is also over. Coming to the omega oxidation. So this omega oxidation is the next step of fatty acid metabolism, and this omega oxidation of fatty acids occurs in endoplasmic reticulum. That is a major difference, and there is another pathway in some species, including the vertebrates, that involves oxidation of omega carbon. You can see here, okay, in the previous reactions I already shown to you, the beta oxidation happens in the beta carbon. Okay, and now the omega oxidation is going to occur in omega carbon. So that is the difference. Here this is the omega carbon. Okay, the carbon is called as omega carbon. So this omega carbon, you can see here mixed function oxidase, and this oxidase utilizes NADPH and oxygen, also atmospheric oxygen, and NADPH with oxygen and converting CH3 to CH2OH, and alcohol dehydrogenase is a very peculiar enzyme, and this alcohol dehydrogenase converts this CH2OH into CHO, this alcohol to aldehyde. Very simple, dehydrogenase, and NAD to NADH will be formed, a removal of one hydrogen, and this aldehyde is aldehyde dehydrogenase, the same enzyme, okay, further removes, okay, one hydrogen and forms aldehyde to, okay, acid. Okay, this is acid. Aldehyde, removal of one hydrogen, NAD to NADH, and the remaining is COO. Okay, this is called as acid. And this acid, okay, they're having two acid moieties. Okay, the one in the first carbon and another in the second carbon, the last carbon, that is omega carbon, undergo beta oxidation and removes a succinate and also adipate as end products. So this is called as adipic acid and succinic acid. Okay, and these are the end products of that is omega oxidation, and I'll explain about, okay, this one, the enzymes unique to omega oxidation are located in endoplasmic reticulum of liver and kidney. So in animal cells, the endoplasmic reticulum of liver and kidney are actively participating in omega oxidation, and the preferred substrates are fatty acids of 10 to 12 carbon, not 16 carbon and all, 10 to 12 carbon only. And the first step involves the hydroxyl group of omega carbon by mixed function oxidase involved in cytochrome P450 and NADPH. Okay, this oxygen is mediated by cytochrome P450 mediated reaction and produce a carboxyl group at end, that is each end. That is, you can see here, this is one end and another end, both ends having carboxyl moiety, and in each pass through the beta oxidation pathway, the double-ended fatty acid yield dicarboxylic acid such as succinic acid, which enter into the citric acid cycle. Okay, so this is succinic acid, succinate or succinic acid, both are same, and this will enter into the TCA cycle. The end product of omega oxidation is either adipic acid or otherwise succinic acid. So next, coming to the alpha oxidation of fatty acids, and so this is omega oxidation and this is alpha oxidation. Alpha oxidation means oxidation at alpha carbon. So I will show it again. This is alpha carbon. You can see here, this is beta oxidation at beta carbon, and this is alpha oxidation at alpha carbon. Okay, we can see here. And alpha oxidation, the branched chain 20 carbon fatty acid, that's called as phytanic acid. And this is not a substrate for acetyl-CoA dehydrogenase because the methyl group of beta carbon. Okay, instead it is hydroxylated at the alpha carbon by phytanyl-CoA alpha hydroxylase, and carbon 1 is released as carbon dioxide. I'll explain. Okay, in this path, you can see here this. So this is the first alpha carbon, the phytanic acid, a branched chain fatty acid. This will undergo the complete oxidation process and releases each and every time. Okay, this acetyl coenzyme A. Okay, the carbon 1 releases carbon dioxide as end product, and 19 carbon pristanic acid is activated to its coenzyme A derivative and undergo beta oxidation. So that means the first carbon is released in the form of carbon dioxide, alpha carbon, and remaining things are undergo beta oxidation. 19 carbon pristanic acid undergo beta oxidation, and this is 19 carbon, students, that is odd number. So 19 carbon minus 3 means approximately 16, C16. C16 divided by 2, that means 8 numbers of acetyl coenzyme A formed, and remaining propanol CoA undergo the further steps. Okay, like this one I already discussed about this one. This is propanol CoA 3 carbon, and this will form succinyl CoA and enter into the TCA cycle. So this is the fate of alpha oxidation, and this phytanic acid is a very important example, okay, for that. And this is called as phytanic acid, a branched chain fatty acid. So with this we can conclude alpha oxidation, omega oxidation, and beta oxidation. Next, coming to the ketone bodies, and we will see in the next session. And with this we can complete all the beta oxidation, alpha oxidation, and omega oxidation. So that is a degradation of fatty acids. Thank you.